Mitosis is a type of equational division in which chromosomes replicate and become equally distributed among two daughter nuclei. The phenomena was first observed in plant cells by Strasburger in 1870 and in animal cells by Bowry and Fleming in 1879. And the term mitosis was coined by Fleming in 1882. Mitosis is a usual method of cell division in all eukaryotic organisms. It is characterized typically by resolving the chromatin of the nucleus into a thread-like form, which condenses into chromosomes, each of which separates longitudinally into two parts called chromatids. Each of these chromatids is retained in each of the two daughter cells resulting from the original cell. The development of an individual from zygote to adult stage takes through mitotic cell divisions, and it is the most common method of division which brings about growth in multicellular organisms and increase in population of unicellular organisms. Therefore, mitosis is necessity for maintenance and perpetuation of life. One of the basic requirements of cell division meant for growth would be that it should give rise to two daughter cells which should resemble each other and also the parent cell both qualitatively and quantitatively. Since a cell also has to maintain continuity from one generation to another, and since the hereditary material within the chromosomes has to copy itself most faithfully, a cell has to divide in such a manner that the two daughter cells are similar to each other and resemble the parent cell from which these were produced. The basic outline of cell division in all the living organisms is almost same, which is divided into different phases constituting what's called a cell cycle. Cell cycle can broadly be divided into interphase and mitosis. In the interphase, cell prepares materially for the division, and the mitosis or M phase is main cell division phase where cell actually divides into two. Let's analyze the events which take place during interphase. Interphase is a phase of cell cycle in which the cell spends most of its time and prepares itself for cell division besides performing normal cellular activities. In this preparation, the cell increases its size, centrioles divide, proteins are actually produced, and more importantly, makes a copy of its DNA. The interphase is divided into three sub-stages. G1 or first growth phase. It is the phase in which the cell grows and functions normally. During this time, much protein synthesis occurs and the cell grows to about double its original size. More organelles are produced, increasing the volume of the cytoplasm. There is a restriction point present at the end of G1 phase. This checkpoint is actually a series of safeguards to ensure that the DNA is intact and no repairment is required and that the cell is functioning normally. Functionally, these safeguards exist as proteins known as cyclin-dependent kinases or CDKs. This checkpoint is also called S-phase promoting factor SPF or G1 S-phase checkpoint. A cell may pause in the G1 phase before entering the S phase and enters a state of dormancy called the G0 phase. Most mammalian cells of neuro and muscle tissue systems do this. Cells enter the G0 phase from a cell cycle checkpoint in the G1 phase, such as the restriction point as in animal cells or the start point as in yeasts. This actually occurs in response to lack of growth factors or nutrients. During the G0 phase, the cell cycle machinery is dismantled and cyclins and cyclin-dependent kinases disappear. Cell then remains in the G0 phase until there is a reason for them to divide. Some cell types in mature organisms, such as parenchymal cells of the liver and kidney, enters the G0 phase semi-permanently and can be induced to begin dividing again only under very specific circumstances. Other types of cells, such as epithelial cells, continue to divide throughout an organism's life and rarely enter into G0 stage. Now we come to S phase, or synthetic phase of cell cycle. This is a phase in cell cycle in which the synthesis of DNA via semi-conservative replication takes place. The DNA, as we know, is the brain of the cell. Hence, the cell will need to copy its DNA faithfully in order to pass it on to two daughter cells. At the beginning of the S stage, each chromosome is composed of one coiled DNA double helix molecule. The enzyme DNA helicase splits this DNA double helix molecule, and DNA polymerase starts attaching complementary base pairs to the DNA strand, making two new semi-conservative strands. At the end of this stage, each chromosome has two identical DNA double helix molecules and therefore is composed of two sister chromatids which remain joined at the centromere. The centromere is also duplicated at this stage. The end result is that duplication of genetic material in the cell, which will eventually be divided into two. Damage to DNA often takes place during this phase, and DNA repair is initiated following the completion of replication. Incomplete or poor DNA repair may flag cell cycle checkpoints, which halts the cell cycle. However, after the cell has completed this phase, it is very likely that the cell will continue to complete the cell cycle. Now G2, or second growth phase in the cell cycle. It is a phase in which cell undergoes a period of rapid growth. It is the last stage of the cell cycle up to which nucleus is well defined, enclosed by nuclear membrane and with nucleolus. Chromosomes, although replicated, are in the form of loosely packed chromatin fibers. As in G1 phase, at the end of this phase 2 is a control checkpoint called G2M checkpoint. The duration of various phases of cell cycle vary from organism to organism and even in different tissues of the same organism. Cells in growing animal embryos can complete their cell cycle in less than 20 minutes. The shortest known animal nuclear division cycle occurs in Drosophila embryos, which is about 8 minutes. Most cells of the adult mammals spend about 20 hours in interphase, and this accounts for about 90% of the total time involved in cell division. However, generally, if we assume the cell cycle of a particular cell to be of 24 hours, more than 23 hours of it will be consumed in interphase, and only less than 1 hour will it spend in mitosis or M phase. The onset of mitosis is followed by the formation of the mitotic cyclin CDK complex, known as M phase promoting factor, that occurs as a cell cycle regulatory mechanism in the G2 phase. The mitosis or M phase of the cell cycle, although a continuous process, is for the sake of convenience and understanding divided into following subphases. Pre-prophase. In plant cells only, prophase is preceded by a pre-prophase stage. In highly vacuolated plant cells, the nucleus has to migrate into the center of the cell before mitosis can begin. This is achieved through the formation of a phragmosome, a transverse sheet of cytoplasm that bisects the cell along the future plane of cell division. In addition to phragmosome formation, pre-prophase is characterized by the formation of a ring of microtubules and actin filaments called pre-prophase band. underneath the plasma membrane around the equatorial plane of the future mitotic spindle. This band marks the position where the cell will eventually divide. Prophase. At the beginning of the prophase, the nucleus of the cell becomes sapphiride, viscosity of the cytoplasm increases, and the chromatin fibers start condensing and thinning to take the shape of chromosomes. Chromosomes become coiled, shortened, and more distinct as the prophase progresses. This shortening and thickening of chromosomes is due to two reasons. Number one, coming together of scaffolding or axial proteins, and number second, lateral looping and coiling of chromatin fibers, which is assisted by a category of proteins called condensins. At the early prophase, the chromosomes are evenly distributed in the nucleus. As the prophase progresses, the chromosomes begin to align themselves to the periphery of the nucleus, creating a clear central area. Chromosomes continue shortening and thickening to assume the characteristic shape and size which is necessary for their equitable distribution later. An important and characteristic feature of the prophase is longitudinal splitting of each chromosome into two sister chromatids, which remain attached to each other only at centromere. In the late prophase, chromatids at the centromere will get attached with the spindle fibers with the help of kinetochore. The nucleolus and nuclear membrane, with some exceptions, start disappearing. The centrosomes, which are the organizing centers of microtubules, begin to separate to opposite poles of the cell. The major event making the cell's entry into pro-metaphase is the breakdown of the nuclear envelope into small vesicles. Distinction between cytoplasm and nucleoplasm fades, and the organelles like endoplasmic reticulum and Golgi apparatus disorganize. The segregation of the replicated chromosomes is brought about by a complex cytoskeletal machine with many moving parts, the mitotic spindle. It is constructed from microtubules, mostly tubulin dimers, and their associated proteins, which both pull the daughter chromosomes forward towards the poles of the spindle and move the poles apart. Microtubules emerging from the centrosomes at the poles of the spindle reach the chromosomes. The attachment of the chromosomes with the spindle is a dynamic process. It seems to involve a search and capture mechanism in which microtubules radiate from each of the rapidly separating centrosomes, grow outside towards the chromosome. In the spindle, some microtubules extend from one pole to the center of the cell, where their ends overlap with the ends of other microtubules that extend from the opposite end of the spindle. The two sets together overlapping in the center form a large framework. Other microtubules run from a pole to a centromere. Each end of the spindle is attached to one of the two faces of the centromere at kinetochore on each chromosome. About 15 to 35 microtubules attach to each kinetochore. The role of prometaphase is completed when all of the kinetochore microtubules have attached to their kinetochores, upon which metaphase begins. Now metaphase. Once the chromosomes get captured by the microtubules of the spindle, the microtubules push and pull on the chromosomes, gradually aligning them to the cell center or spindle equator, forming what is called a metaphase plate. This alignment is due to the counterbalance of the pulling powers generated by opposite kinetochores. Mitotic cells usually spend about half of M phase in metaphase, with the chromosomes aligned on the metaphase plate, jostling about, awaiting the signal that induces sister chromatids to separate to begin anaphase. This phase is marked by a checkpoint called spindle checkpoint. The checkpoint mechanism is used by the cell cycle control system to ensure that the cells do not enter anaphase until all the chromosomes are attached to both poles of the spindle. When every kinetochore is attached to a cluster of microtubules and the chromosomes have lined up along the metaphase plate, the cell proceeds to anaphase. During anaphase, the kinetochore microtubules retract, increasing the separation of the sister chromatids as they are moved further towards the opposite spindle poles. Anaphase can be subdivided into two distinct phases. In the first phase, called anaphase A, chromosomes move poleward away from the metaphase plate with the retraction of the microtubules. This movement occurs at approximately 2 micrometers per minute. In the second phase, or anaphase B, the mitotic poles marked by the centrosomes themselves separate by the elongation of a specific type of non-kinetochore microtubule called a polar microtubule. The entire duration of the anaphase is relatively short, usually only lasting for a few minutes. As if following a neatly choreographed dance, the sister chromatids separate, rapidly moving towards a pole to which their microtubule is attached. The cell appears stretched as the spindle fibers slide past one another, elongating the spindle apparatus and further separating the poles. Shortening of the microtubules by removal of tubulin units pull the chromosomes closer and closer to the pole. The movement of sister chromatids to opposite sides of the cell completes the equal division and distribution of genetic material. The amount of energy necessary to move a chromosome from the metaphase plate to end of the spindle is small. Just 20 ATP molecules are sufficient. Long chromosomes may tangle somewhat, but microtubules exert sufficient pull to untangle them and drag them to the ends of the spindle. Because the spindle is shaped like a football, as chromosomes on each side get closer to the end, they are pulled together into a compact space. Chromatids separate from each other with the breakdown of the cohesion linkage, the protein complex that binds the sister chromatids at the metaphase plate. This metaphase to anaphase transition is triggered by the activation of anaphase promoting complex or cyclosome APC. The APC is a complex of several proteins. It is the last nuclear division phase in which the spindle apparatus disintegrate as the microtubules are broken down into tubulin monomers that can be used to construct the cytoskeletons of the daughter cells. Fragments of nuclear envelope appear near around each set of the sister chromatids, which can now be called chromosomes, connect with each other and form complete nuclear envelope around each set of chromosomes. The chromosomes again short, uncoiling into more loose threads of chromatin, which enable them for genetic expression. Gradually, new nucleoli appear as the ribosomal genes become active and produce ribosome subunits. The spindle depolymerizes completely and disappears. Most of the events in telophase are the reversal of those in prophase. Now we come to cytokinesis. Cytokinesis is the process in which the cell actually divides into two. With the two nuclei already at opposite poles of the cell cytoplasm separates and the cell pinches in middle, ultimately leading to cleavage. Cytokinesis is technically not even a phase of mitosis, but rather a separate process necessary for completing cell division. In animal cells, a cleavage furrow containing a contractile ring develops where the metaphase plate used to be, pinching of the separated nuclei. This contractile ring, which is made up of myosin second and actin filaments, assembles in the middle of the cell. Myosin second move along these actin filaments, constricting the cell membrane to form a cleavage furrow. Continued hydrolysis causes this cleavage furrow to move inwards. Ingression continues until cell is finally cleaved into two. In plant cell, due to the presence of a cell wall, cytokinesis is significantly different from that in animal cells. Rather than forming a contractile ring, plant cells constrict a cell plate in the middle of the cell. The Golgi apparatus release vesicles containing cell wall materials. These vesicles fuse at the equatorial plane and form a cell plate. The cell plate begins a fusion tube network, which then becomes a tubulovascular network TVN as more components are added to it. The TVN develops into a tubular network which then becomes fenestrated sheet which adheres to the existing plasma membrane. In both animals and plant cells, cell division is driven by vesicles derived from the Golgi apparatus which move along microtubules to the middle of the cell. A cell uses three checkpoints during the whole process at specific points in the cell cycle to assess the accuracy and to integrate the external and internal signals before its transition to next phase. These checkpoints are G1S checkpoint. This is the first checkpoint where a cell, in response to surroundings, can decide whether to divide or not. The possible factors which govern this checkpoint include growth factors, nutritional status of the cell, and the fitness of the genome. Poor nutritional status, lack of growth factors, and damaged DNA can halt the cell cycle at this point. This checkpoint has been well studied in yeasts, where it is being termed as start. In animals, it is termed as restriction point. Now G2M checkpoint. This is the second and most important checkpoint which is meant for assessment of DNA which has been synthesized in S phase. This checkpoint is commonly referred to as mitosis promoting factor MPF. The G2M checkpoint provides an opportunity for repair of damaged DNA if any, hence stopping the proliferation of damaged cells, therefore help to maintain genomic stability. Passage through this checkpoint represents the commitment to mitosis. Now spindle checkpoint at the metaphase. This checkpoint comes into action just before the partition of two sets of chromosomes destined for two daughter cells at metaphase. The second irreversible step in cell cycle is the segregation of chromatids during anaphase. Hence, the spindle checkpoint ensures that all the chromosomes are attached to spindle fibers correctly before transition into anaphase.